My name is Samantha Lumley. I am in Bio 225 and I am doing video for Aseptic Technique. It is May 15th, 2019, and I had already done the first part of the experiment, but I'm going to do a short instruction on how I did the first part. So it called for alcohol and for a candle and a pipette and broth. Here's the broth, nutrient broth. I labeled it on the back. S. Cerevisiae, which is the yeast. For example, the yeast had me put it in a cup with water and let it dissolve. Mix the cup, let it dissolve and sit for 10 minutes. Also, it called for a candle. So, and then the cup of alcohol in the pipette. I'm going to expel the alcohol from the pipette. Make sure it's dry. So, then I drew up 0.25 milliliters in here. And then I had opened up the nutrient broth. You pass the top of it over the candle to sterilize. I took the 0.25 and I inserted it into, or dropped it into the nutrient broth without touching the edges. Then I recandled the edge and capped the nutrient broth. I expelled the yeast, put it into the alcohol, drew it up all the way into, and I did that for E. coli and I did that for the Staphylococcus epidermidis. Those were tablets, so they were in little canisters and mixed with water, or the nutrient broth, sorry. It was mixed with the nutrient broth, shook around, dissolved the tablet, drew it out, and dropped the dissolved tablet into the nutrient broth. So that was the first part of the experiment. Hi, my name is Samantha Lumley. I am in Bio 225. It is May 15, 2019, and I'm doing the second part of the experiment, streaking the plates. I've already done 2. I just donned gloves and I've got my plates down here, and there's the alcohol cup. These are the plates that I just streaked, as labeled it on the bottom of the agar plate. These are the broth tubes that I have created. So this is the last one. This is the alcohol cup. This is the yeast. I'm going to first open it up, pass it over the flame. Pass it over the flame. I am drying off the loop without touching the edge. I'm going to insert and touch the bottom. Take it out. Now I'm going to streak the first quadrant of the plate. Back and forth, streaking the first quadrant. Then it goes into the alcohol for 20 seconds. I cover the plate up. 1,000, 2, 1,000, 4, 1,000, 5, 1,000, 6, 1,000, 7, 1,000. I take it out, I tap it. I'm not touching the loop on anything, it's just in the air. I'm just tapping the edge of the handle of the loop. Then I'm going to do the second quadrant, crossing over the first quadrant. and then it goes back in, and I cover it up. 20 seconds. 9,003. 11, 12, 13, 14, 20. Okay. Then I tap it dry. Okay, it's dry, and then I cross over the third quadrant. Make sure I cross over the second quadrant. Streaking the plate. And then 20 seconds in the alcohol. 1,006, 1,000, 1,010, 1,000. Okay, and then I tap it dry. I open up the last quadrant. 1, 2, 3, and then I cross over the first quadrant. There we go. Being careful not to cross over. I cross over the third quadrant. Being careful not to cross over the first quadrant. Sorry. Okay, then I cover it up. I turn it upside down, and they're all labeled on there, and that is it. Then I'm going to flame the lid of the yeast vial and close it up, and then I'll be storing it in the refrigerator for next assignment. And that is on the E. coli, there's only a few that actually grew on here. To look at them and describe them, there are, for the S. epidermidis, they are circular, and I have my description right here sheet to help me remember some of the terms. But they are circular, and they do have a margin. They are not flat. They look like they are convex, and the size of them looks like the really separated ones are pinpoint size, and then the group, it looks like the group of microbes are like a large grouping of them. On the yeast, it's the same thing. They're circular and raised, like convex. They are, the margin has an entire margin. The size on the ones that are separated are pinpoint, and then the colonies, there's like a microbe colony, they're large, they're pretty round, moderate size, small to moderate. And then the E. coli, there's only a few, and they have like this, they do have circular shape, and they're pretty, in the margins entire. The elevation looks convex as well, and the size is small to punctiform. So, and there's only 4 of these guys on there, unless these like really tiny pinpoint ones are separated right here. which then the streaking method probably did work on this one too, but just not as many microbes. So that is my lab today for the aseptic technique in culturing microbes. Thank you.